It had become an annual ritual. A small group of graduate students from the University of Utah would accompany their advisors to the Wasatch Mountain Ski Resort of Alta for an intensive workshop on their research and, well, a little skiing on the side. Typically, a couple of big-shot scientists from other institutions would be invited to cast a critical eye over the data presented by each nervous student. In 1978, the big shots included David Botstein from MIT and Ron Davis from Stanford. David Botstein, it's been noted, tends to think and talk excessively fast, and often at the same time. Ron Davis is quiet and retiring. That April in Utah, despite their contrasting styles, Botstein and Davis shared an epiphany. As they listened to Mark Skolnick's graduate students discuss genetic disorders, traced in the very large pedigrees of Mormon families, Botstein's and Davis's eyes suddenly met as both registered, simultaneously, the same insight. Though both were experts on yeast, they saw a way to locate human genes. What they saw was that cutting-edge recombinant DNA techniques would allow them to apply to humans the very sort of genetic analysis first used by Morgan to study the fruit fly. In fact, DNA markers had already been used to map genes in a number of other species. But Botstein and Davis would be the first to develop the technique's potential in humans. The technique, called linkage analysis, determines the position of a gene in relation to the known positions of particular genetic landmarks. The principle is simple. It would be difficult for you, given no other information, to find Springfield on a map of the United States. But if I tell you that Springfield lies about halfway between New York and Boston, two landmarks labeled on the map, then your task is made very much easier. Linkage analysis aims to do this with genes. It establishes links between known genetic markers and unknown genes. It was a very successful method with the fruit fly. But, as we have seen, the dearth of known genetic markers in human beings prevented its application to human diseases, until Botstein and Davis recognized that advances in molecular biology had solved the problem. The DNA markers that caught their eye were restriction fragment length polymorphisms, RFLPs. They occur when a DNA sequence cut by a particular restriction enzyme in one individual has changed in another, so that it can no longer be cut by that enzyme. Remember that restriction enzymes are sequence-specific. Enzyme EcoR1 cuts only when it encounters GAATTC. That sequence occurs at a given location in the genome, but through mutation, some individuals may have a variant form of that segment, say GAAGTC. The enzyme will be able to cut only unchanged sequences, not the altered version. These are naturally occurring differences in DNA sequence. They occur most often in junk DNA, and so there is no functional effect. Still, literally millions of them are scattered through our genome. In the months following the Alta meeting, Botstein, Davis, and Skolnick, together with Ray White, then at the University of Massachusetts, pursued the RFLP concept. In 1980, a landmark paper that grew out of this collaboration heralded the new age of molecular human genetics. They laid out a clear plan showing how RFLPs could be used, and they worked out the math concerning how many would be needed to ensure that every point in the human genome was within reasonable proximity of at least one RFLP marker, conditions that would, in principle, permit the mapping of the entire genome. It would be like having enough U.S. cities fixed on a map of North America to allow any unmarked place to be located with respectable accuracy, using only information about how close it was to the labeled cities. But for the genetic map, what was reasonable proximity. Botstein and his colleagues calculated that 150 RFLPs spread uniformly across the entire human genome would be enough. The most immediate benefit of the system was a new strategy for identifying genes that cause disease. Using families in which a disorder spans several generations, they would take DNA samples from both affected and unaffected individuals. Then they would use recombinant methods to test RFLPs, one after another, looking for ones that tracked the disease through the families. In 1979, before the publication of the paper, White presented these ideas at a Cold Spring Harbor Laboratory conference. He noted that among the more kosher molecular biologists, there was a lot of bitching and grumbling. What he was hearing was great skepticism as to whether the method would work at all. Even those who thought it would couldn't agree on the best way to go about using it. These disagreements came into the open during a later meeting to discuss how RFLP linkage analysis could be used to find the gene involved in Huntington disease. Nancy Wexler wanted her Lake Maracaibo genealogy to be considered immediately for linkage studies. But Botstein and White thought it was far too early to use RFLP linkage analysis to look for the Huntington gene, or any other. They argued that much groundwork needed to be done first. The markers themselves had to be found and mapped before the technique could be applied for such a specific purpose. In the end, Wexler's determination resulted in a parting of ways. While the Hereditary Disease Foundation pressed on with the hunt for the Huntington gene, Botstein and White pushed for a complete map of the human genome. The latter goal required finding RFLP markers on every chromosome, and finding enough of them to ensure that at least one was close to every point in the genome. It was soon necessary to make an upward revision of the initial estimate of 150. But undeterred, academic laboratories like White's began to isolate RFLPs, and soon commercial biotechnology was getting in on the action as well. In 1983, Helen Donis Keller, an experienced molecular biologist and in those days David Botstein's wife, established the Human Genetics Department at Collaborative Research, Inc., a Boston area company.
This book is continued on Disc 11.